Good evening, everyone, and welcome to Futures webinar series. I'm Ivana Jeftović, webinar coordinator. Mosquito-borne diseases or mosquito-borne illnesses are diseases caused by bacteria, viruses, or parasites transmitted by mosquitoes. Nearly 700 million people get a mosquito-borne illness each year, resulting in over 1 million deaths. How have the scientists fought against these health problems? What goes on behind the scenes in labs? Dr. Jovana Božić will lead us through the work of her team in search of new knowledge and strategies on how to recognize, prevent, and control vector-borne diseases, such as those transmitted by mosquitoes. Dr. Jovana Božić is a postdoctoral fellow at the Florida Medical Entomology Laboratory at University of Florida. Jovana completed her bachelor's at the University of Banja Luka in Bosnia and Herzegovina. She moved to Italy to complete her master's degree and at the International Center for Advanced Mediterranean Agronomic Studies. After her master's degree, Jovana focuses on in research shifted from integrated pest management and agricultural crops to vector biology and mosquitoes. Jovana completed her PhD in environmental sciences and public health, Malaria and Human Development. At the University of Camerino, Italy, as part of her PhD, Jovana was also researcher scholar at the Yale School of Public Health. After her PhD, she completed one postdoc position before joining the Southeastern Center of Excellence in Vector Borne Diseases. Jovana focuses on research for microbial symbiosis in mosquitoes and its influence on mosquito psychology and arboviruses transmissions. Welcome, Jovana. It's a pleasure and honor to have you here. I thank you in the name of Bosnia and Herzegovina Futures Foundation, and I give you a word now.
Thanks, Ivana, for the introduction. Hello, everyone. I'm Jovana again, and today I will talk about mosquitoes and their association with different microorganisms, and also about novel approaches to control mosquito-borne diseases. In this presentation, I will walk you through my academic journey all the way from the very beginning. I will briefly summarize my undergrad and master projects, and I will mostly focus on my PhD and current postdoc research. I completed my bachelor's at the University of Banja Luka, Bosnia and Herzegovina, Faculty of Agriculture. There I studied pomology, viticulture, and pests and diseases of fruit tree crops and grapes. After I completed my bachelor in 2009, I started application for a master program, as I recall well, at the plant protection at the Mediterranean Institute of Bari. Dr.Jovana Božić from the University of Banja Luka, who was a master fellow there, has recommended me for this program. And actually, this was a huge step for me, one of the crucial milestones, I can say, in my career. And therefore, I couldn't be more grateful to Jovana for putting an effort to help me in this process. More acknowledgements will be mentioned at the end of the talk. I moved to Italy to complete my master's degree at the International Center for Advanced Mediterranean Agronomic Studies, Mediterranean Agronomic Institute of Bari. My master research focused on integrated pest management of Mediterranean fruit tree species, specifically palm pest, the red palm beetle, Rhynchophorus ferruginellus. From this research, I gained an interest in pest management and entomology. After the master's degree, my focus in research shifted from integrated pest management and agricultural crops to vector biology and mosquitoes. I started PhD in environmental sciences and public health with the focus on malaria and human development at the University of Camerino in Camerino, Italy. On the slide, you can see this wonderful city. As a graduate student and European Research Council Fellow in the Parasitology and Medical Entomology Lab, I was mentored by Dr. Irene Ricci, coordinator of the project, and supervised by Dr. Guido Favia, who was and he still is head of the lab. My dissertation research looked into the characterization of fungal symbiosis in vector mosquitoes and the evaluation of its potential impact in the control of vector-borne diseases. Specifically, I investigated interactions between the mosquito host, fungal symbionts, and malaria parasite at the molecular level, and also I was working on the manipulation of fungal symbionts expressing antipathogen molecules within the mosquito host. What does it actually mean? I will describe in the following slides, which are all about mosquitoes. So let's start. I will initiate my mosquito story by introducing some terminology which will be helpful later in the description of the projects I work on. Vector-borne disease is a disease that results from an infection transmitted to humans and other animals by blood-feeding arthropods such as mosquitoes, ticks, and fleas. Efforts to control vector-borne diseases have been compromised by the evolution of insecticide resistance and failing drug therapies. As you can see, I showed those schemes on the slide. To improve the sustainability and efficacy of control efforts, alternative vector control strategies are being considered, like methods that suppress the pathogen instead of the vector. This is the part where actually insect symbiosis comes on the scenes. Symbiosis in general usually refers to all cases where different organisms or species, for example, heterogenetic associations, live together in a close association. Symbiotic microbes can provide a number of functions to the host, to the insect host, and allow the insect a great ability to adapt to the environment. Symbiotic associations have been described in insects that rely on nutritionally restricted diets, such as vertebrate blood, like tsetse fly and ticks. It will be a little bit talk about that later. Or on a diet that requires blood besides feeding on a flower nectar, like in the case with mosquitoes. On this slide, I presented 4 mosquito species relevant to public health. They are also in specific association with different microbes which help them to maintain their overall well-being. In addition to carrying beneficial microbes that enhance their overall physiological functions, some insects also carry and transmit microbes that are pathogenic to their human, animal, or plant host. Most of the insects, including mosquitoes, which are disease vectors of humans, utilize blood as a food source and typically acquire pathogens while feeding on infected hosts and then pass the disease agents onto other hosts during the course of subsequent meals. Many vector insects exhibit a native resistance to the pathogens they transmit, and that resistance comes from beneficial microbes. A part of that resistance actually comes from their beneficial microbes, which are called symbionts, gut microbes, or in general, mosquito microbiota or insect microbiota. I will describe this interaction using the example of malaria parasite and arboviruses. On this slide, you can see infection of malaria parasite in Asian malaria vector Anopheles stephensi. So malaria infection begins when infected female mosquito bites a person, injecting Plasmodium parasites into the bloodstream. When a mosquito bites an infected person, it ingests the gametocytes, one of the developing stages of the malaria parasite, which further develop into the mature sex cells called gametes. However, once the parasites reaches the mosquito gut, it has to overcome several bottlenecks. So gamete formation requires parasite to exit from the enveloping red blood cells, rendering them susceptible to aggressive factors of the insect gut, like for instance gut microbes or the components of the blood meal. Further on, the fertilized female gametes develop into actively moving ookinetes. So this is actually like active stage of the parasite. that burrow through the mosquito midgut wall and form oocysts on the exterior surface. The replication phase requires roughly 2 weeks and results in the formation of infective sporozoites that migrate to the salivary glands to be released into the human skin with the next bite of the mosquito, wherewith the life cycle of parasite is completed. The second crucial moment takes place between the midgut epithelium and basal lamina, where oocysts rest. It has been reported a significant reduction in the parasite at this life stage, considering of the tens of thousands Plasmodium gametocytes, malaria parasite gametocytes, actually particles ingested by mosquitoes through the blood meal. So it has been calculated that less than 5 oocysts are produced, which means that mosquito has its own immune response and means of fighting this pathogen. Regarding the arboviruses, the female mosquito Aedes, which is other mosquito species responsible for virus transmission, feeds on virus-infected blood meal and then occurs so-called ectristic incubation period in the vector. So actually, virus has to become infectious, and that's why actually it's called incubation period in the vector. Virus infects the midgut cells, replicates and disseminates to the hemocell, which is the primary body cavity containing circulatory fluid, and also salivary glands, but also other organs. It penetrates the basal lamina of the salivary glands, and their mosquito saliva is actually stored prior to its release during the feeding. Since the microbial symbionts are localized within the ingested arboviruses, their presence in mosquito gut can decrease viral load in mosquitoes. So mosquito-associated microbes are therefore building blocks for alternative control strategy defined as symbiotic control. Symbiotic control proposes the use of symbiotic microorganisms which inhabit the gut of the mosquito as a Trojan horse to eliminate the parasite from mosquitoes and thus block its transmission in humans. Symbiotic control have developed a strategy so-called paratransgenesis, which is the process of genetically transforming mosquito symbionts to confirm specific functions that decrease vector competence to pathogens. So this is on your left, you can see very simple scheme of the main aspects of symbiotic control. So first of all, first step in this process is isolation of symbionts from the insect, activation of the symbionts for the expression of antiparasitic molecules. It's usually made through genetic manipulation. Reintroduction of the activated symbiont into mosquito and elimination of the parasite. In order for, on your right side, actually, it's also the same image, resembles the same concept, but this is for, I put this on purpose for the people who are more into molecular biology. In order for paratransgenesis to be successful, a symbiont must successfully associate with the vector in the field. It has to be genetically tractable. It doesn't have to pose harm to the environment. Also, doesn't have any fitness cost to the vector, and also has to be amenable to the genetic transformation and able to colonize vector population in vitro or in vivo. So during my PhD, I was working on yeast symbiont associated with malaria vector, and I was mostly working on its genetical engineering. This yeast was isolated from the gut of Asian malaria vector, Anopheles defensa, and actually it was a milestone discovery from my professor Irene Ricci. The yeast was called Vickeromyces anomalus. This yeast is particularly interesting because it's widespread in the nature. It is isolated from different matrices like environment. It's isolated from insects other than mosquitoes, and it's also found in immunocompromised patients. It shows antimicrobial activity through the killer toxin production, and also it's amenable to genetic manipulation. It has all the attributes to be perfect candidate for symbiotic control. Contribution to the knowledge of yeast symbiosis in mosquitoes, mainly for basic and applied studies, is the #1 significance of this research. Green fluorescent protein, GFP taxane, will permit to precisely localize the yeast in the mosquito organs and to follow its spreading dynamic within and across mosquito population in case semi-field or field studies. So actually GFP is a green fluorescent protein, is a marker protein, and it can attach and mark another protein with fluorescence, reporting in this case localization of yeast in mosquito body. So actually by enhancing natural antimicrobial properties of this symbiont through genetic engineering and increasing the expression of killer toxins, this study could contribute the efforts to reduce vector competence of mosquito and pathogen transmission. Unfortunately, these projects stay unpublished because many problems, we encountered many problems during the engineering experimentation. However, this is my to-do list, and actually this technique can be applied also to other mosquito-associated yeasts. As I mentioned previously, yeast symbionts shows antimicrobial activity by producing killer toxins, and this has been tested against different stages of malaria parasite, and this was also tested in vitro. Alterations observed in killer toxin treated parasite using yeast sustaining confirmed that killer toxin produced by yeast symbiont targets the developmental stages of the parasite. If you can see, the treated version is from D to F, and untreated is from A to C. So you can see the differences in the shape of the one of the parasite stages. Usually, normally it's like in the C, but when it's treated with a killer toxin, it becomes totally destroyed. So I also put malaria cycle again on this slide. I don't know how much it is visible. So you can actually clearly see which stage of the parasite is, of parasite growth is blocked in this experiment. So to repeat, those are the okinids, and okinids are the bugs that burrows the mosquito midgut wall and form oocysts, which later reach salivary glands when they burst into sporocyte, which is equal infected mosquito. This interesting study was published 2016 in Malaria Journal, and I was one of the co-authors on this study. So going further through my PhD, I studied different fungal symbionts. Among these, also the opportunistic pathogens. Opportunistic pathogens, by definition, are typically characterized as organisms that can become pathogenic following a perturbation to their host. Having said that, the other yeast that we isolated from mosquitoes at the University of Camerino is the yeast Candida parapsilosis. Is known as a human commensal, but recently it has also been identified as an opportunistic pathogen and in immunosuppressed or immunocompromised patients. Mosquitoes, since they feed on a blood So they are hematophagous insects. They can play a role in the dissemination of these opportunistic pathogens among humans and domestic animals. So in this descriptive research paper published in Environmental Microbiology Reports in 2016, the stable association of opportunistic yeast pathogens, opportunistic yeast and mosquitoes is described, and actually how mosquitoes participates in the spreading of this opportunistic pathogen is also discussed. I had one-time opportunity to work at Yale University, Yale School of Public Health, in the laboratory of Professor Serap Aksoy. I carried out screening for the yeast present in another vector, which is tsetse fly, vector of parasite from the genus Trypanosoma, causative agent of sleeping sickness and Nagana disease in animals. Tsetse flies differ very much from other insects, especially they differ very much from mosquitoes, from first of all from a way of their diet, which is consistent solely on vertebrate blood. So they are obligate hematophagous. They also exploit proteins rather than sugars as an energy source, and also their form of reproduction is viviparous. That means, and you can see in this photos on your left, that instead of laying eggs, tsetse develops a single offspring during one reproductive cycle, which is carried and nourished by the mother throughout embryonic and larval development. Tsetse fly accommodates three different endogenous symbionts. Those are bacterial symbionts, but I will not go through all of them. I will just mention the most important one. It's the one in the green. So this bacterium, this bacterium actually provide a nutritional supplementation required for female fecundity and larval development, including provision of vitamin Bs, and also provide immune stimulation for larval system development. In the absence of this bacteria, this insect, during the larval development of this insect, adults are later compromised, severely immunocompromised, and more susceptible to diseases and pathogens. So why tsetse fly? Tsetse was actually interesting course to be examined for fungal symbionts, since like mosquito represent a vector of medical relevance, plus previous studies didn't report tsetse yeast association. Turns out that this vector indeed doesn't harbor any fungal microorganism, which wasn't surprising due to the fly's intimate association with several bacterial symbionts. Internship at Yale was one of my favorite experiences during the doctoral studies. Besides acquiring the new knowledge and tactical practices professionally and personally, I was fortunate to work with amazing people, members of Dr. Serap Aksoy, and I will acknowledge them all at the end of this talk. During my PhD, I was also speaker at the National Congress of the Italian Society of Parasitology in Rome, Italy, presenting the use of symbiotic yeasts associated to mosquitoes to prevent plasmodial infection in malaria vectors. Following the 1 year of postdoc, I was catching up actually research and experiments from my PhD. I also contributed to several publications which were published after I moved to Florida. They're listed on the slide. I was also a lab mentor for undergrad student Elena Quadrini. She was my very, very first student that I followed fully throughout her undergrad lab project. And I'm happy to say that now she's a PhD student at the University of Camerino. So here we are in Florida. Through a connection from my previous research at Yale University, I was connected with Dr. Raul Dinglasan and the Southeastern Center of Excellence for Vector Borne Diseases to begin my postdoc in January 2018 with Dr. Derek Matthias. My research focuses on microbial symbiosis, in mosquitoes and its influence on mosquito physiology and arbovirus transmission. Currently, my work emphasizes two groups of microorganisms. The first is Wolbachia pipientis, a bacterium that I will tell a little bit more in the next slides. And the second group of microorganisms are, of course, yeast, because my fascination with them never ends and I constantly continue to study their interaction with mosquitoes. For this, I also have to thank my supervisor, Dr. Matthias, for allowing me to follow and deepen my interest in this peculiar aspect of mosquito symbiosis. Wolbachia pipientis is an intracellular bacterium that naturally infects a wide variety of insects and also infects multiple mosquito species. It was first reported within the reproductive tissues of the Culex mosquito back in 1924. Wolbachia trigger an array of reproductive manipulations favoring the fitness of infected females over their uninfected males. As I said before, it infects a range of arthropods, insects, and also infects some nematodes. They are possibly the most common endosymbiotic bacteria on the planet, as well as the most common reproductive parasite in the biosphere. I will tell a little bit about those specific phenotypes that Wolbachia induces in insects. So Wolbachia can infect many different types of organs, but are most notable for the infections of the testes and ovaries of their hosts. Wolbachia's preferred habitat is the cytoplasm of its host gametes. Since sperm have very little cytoplasm, Wolbachia infect female cells, securing its survival by moving to the next generation in the cytoplasms of the mother's eggs. Therefore, only infected females pass the infection on their offspring. Owing to this trajectory, Wolbachia have evolved an impressive range of reproductive parasitic strategies to adapt its host physiology to its own advantage. First is male killing, and it occurs when infected males die during larval development, which increases the rate of born infected females. Feminization results in infected males that develop as females or infertile pseudo-females. This is especially prevalent in some butterfly species. Parthenogenesis is reproduction of infected females without males. Some scientists have suggested that parthenogenesis may always be attributable to the effects of Wolbachia. An example of parthenogenesis induced by presence of Wolbachia are some wasp species within the genus Trichogramma, which have evolved to procreate without males due to the presence of this bacterium. Last but not the least, but certainly most importantly, phenotype of Wolbachia is cytoplasmic incompatibility. So cytoplasmic incompatibility is the inability of Wolbachia-infected male mosquitoes to successfully reproduce with uninfected females, leading to embryonic death. In the mechanism of cytoplasmic incompatibility, Wolbachia interferes with the parental chromosomes during the first mitotic division to the extent that they can no longer divide in synchronization. The other aspect of this cross, cytoplasmic incompatibility, occurs in the case when mosquitoes with different Wolbachia types, because there are so many Wolbachia strains in the nature. So when they mate, when the mosquitoes with different Wolbachia type crosses between, the offspring will be sterile. How is this important and how can be implemented as a control strategy? First of all, cytoplasmic incompatibility is one of the phenotypes that can be used to suppress mosquito population through incompatible matings or replace population through the reproductive advantage provided by this mechanism. So population suppression can be achieved through Wolbachia-infected males mating with wild, non-infected females and resulting in cytoplasmic incompatibility or equal no offspring. The objective of the second approach is to replace natural population with mosquitoes possessing Wolbachia infections that can also interfere with the virus transmission. Yes, that's actually very true. Wolbachia can protect mosquitoes from and limit their ability to transmit mosquito-borne pathogen, especially viruses. Aedes aegypti is the principal vector of dengue virus and typically does not harbor this bacterium in nature like many other mosquito species. For this reason, dengue vector Aedes aegypti was artificially infected with this bacterium. On your right, this figure summarizes Wolbachia phenotypes induced by the fruit fly Wolbachia strain infected in Aedes aegypti mosquitoes. So crosses between Wolbachia infected and Wolbachia infected males and Wolbachia non-infected females results in 100% cytoplasmic incompatibility, which is equal to no offspring, while every other crossing combination produced viable embryos. So once an infection is introduced through embryo injection, Wolbachia infections have applications for pest and disease vector control, since they can alter host reproduction and block virus replication and transmission. Therefore, above-mentioned population replacement and suppression methods rely on a novel Wolbachia infections inducing cytoplasmic incompatibility with the resident mosquito population. On the contrary, the presence of Wolbachia in nature, the presence of natural Wolbachia infections in nature and in mosquitoes may interfere with disease control programs, making population replacement or suppression challenging or maybe impossible. So after this long introduction, the main goal of my postdoctoral research here at the University of Florida is to test the hypothesis that Aedes aegypti population from Florida has become infected with Wolbachia symbionts. So far, I can just tell you that this project involves complex molecular and genetic studies, and it will be published soon, so stay tuned for the results. As I mentioned before, I continue to study the fungal microbiota of mosquitoes with an emphasis on yeast acquired by adult females in nature. To this point, I found 9 distinct species of fungi, which are being maintained on agar plates in our lab here at Florida Medical Entomology Lab. My goal is to test their influence on mosquito fitness and vector competence for arboviruses. And this is an ongoing study. As the journey of learning new skills never ends, together with Dr. Matthias and my lab mates, Jody and Tanise, I'm in the process of developing new techniques, studying interactions between the mosquitoes and arboviruses using the Aedes aegypti dengue Zika virus model system. Our lab focuses on experimental approaches to investigating factors influencing arboviral competence in Aedes aegypti populations. I also collaborate with Dr. Chelsea Smart. Dr. Smart is a very acknowledged professor here at Florida Medical Entomology Lab. And I actually in this project, I study the impact of mosquito microbiota on vector competence for dengue virus. Dr. Smart's PhD student, Seiyu Chen, has been working on mosquito immunity that might be disturbed or somehow changed due to the dengue virus infection. My part in this teamwork is to determine whether mosquito symbionts influence this virus-induced stress. Jointly, the results would provide insights into potential factors involved in antiviral responses to develop, again, new strategies for virus transmission blocking. Besides professional collaboration, I also have to say that Dr. Smart and her PhD student, Seiyu Chen, are my dear friends, and I'm very excited for us to publish this work together. So here I will kind of complete my presentation, and these are my take-home messages. So mosquitoes are one of the deadliest animals in the world. Their ability to carry and spread disease to humans causes millions of deaths every year. According to the World Health Organization, in 2015, malaria alone caused around 400,000 deaths. Therefore, sustained and alternative mosquito control efforts are important to prevent outbreaks from those diseases. Mosquitoes carry beneficial microbes that enhance their overall physiological functions and contribute to their overall health. Mosquitoes, like other bacterial insects, exhibit an innate resistance to the pathogens they transmit, which is attributed to their gut microbes that can also stimulate mosquito immune system. And yes, mosquitoes also, like we humans, have their own immune system. Symbiotic control is an alternative strategy that proposes the use of symbiotic microorganisms which inhabit the gut of the mosquito as a Trojan horse to eliminate a parasite from mosquitoes and thus block the transmission of the parasite in humans. Symbiotic control also proposes activation of these microbial symbionts for the expression of antiparasitic molecules. Therefore, symbiotic control has several advantages over current control methods. First of all, it overcomes problems of resistance due to the resistance, which is actually created due to the constant and repeated use of insecticides. It is environmentally friendly. It can be integrated into existing prevention models at a very low cost and also represents an innovative technology that can be easily transportable to endemic regions. And lastly, mosquitoes still serve important functions in numerous ecosystems, serving as food for many species, helping filter detritus from plant life to thrive, pollinating flowers. That is one of the reasons actually why scientists are looking at the mosquito for potential medical treatments. And here I'd like to acknowledge all the members of my bachelor and master committees, with special thanks to Dr. Jovana Božić. Huge acknowledgement goes to my PhD supervisor, Irene Ricci, and Aida Capone. She was actually my teacher, my friend, my mentor, my everything. She was actually a postdoc when I was a student at that time, so she really taught me everything. I would like also to thank all members of my ex-lab in Camerino. I'd like to thank Dr. Ronaldin Glasan and CDC Southeastern Center of Excellence for giving me the opportunity to pursue my academic career as a postdoc at the University of Florida. And finally, many thanks to my lab mates, Jody and Tanise, and our prof, Dr. Matthias, and finally to Dr. Smart and her PhD student, CIHEAM, for their constant support. I also would like to thank to Bosnia and Herzegovina Future Foundation for hosting this webinar and having me as a speaker. It was really great to share my experience with the members of your community. And also thank you all who registered to listen to my talk. Thank you for your patience and attention, and I'm ready to take questions. Thank you.
Okay, I invite everyone to ask questions. Just want to ask you, do you hear me, Jovana?
Yes,
okay.
I can hear you.
Okay, that's fine. We thank you, I thank you in the name of Bosnia and Herzegovina Futures Foundation and in my name. It was really great and inspiring that you mentioned everyone that had some impact in your life and in your career path, and it shows how grateful and how humble human you are, and that's really amazing. I think that you are one of the rarest persons that did this, and that's really amazing. Okay.
Thank you so much, Ivana. Well, I cannot acknowledge everything what I achieved. I cannot attribute that just for myself. Of course, without people and their help and support, and of course, amazing mentors, I wouldn't be in this position. So again, I thank all of them.
That's really
and amazing. Also thank you again.
Yeah, that's really amazing attitude, and you have thanks and greetings from Banja Luka. That attitude is just amazing and inspiring, and you are a great person. So really thank you for this.
You are welcome.
We have one question. So the current methods compromise on the positive effects of mosquitoes.
So the current methods compromise on the positive effects of mosquitoes. You mean in the way that actually mosquitoes still being a little bit beneficial for the ecosystems, pesticide and current methods that are mostly used with the insecticide treatments? I'm sorry, I'm a little bit confused with the question. Okay, yeah, so actually, yes, because mosquitoes, like many other insects, actually can develop resistance to the chemicals. And therefore, like that resistance is a problem because actually once you treat, once you do a large treatment in a wide environment, those mosquitoes slowly will become resistant to pesticides, and also in the same time you will have also some environmental impact from those from those pesticide treatments. Therefore, like those alternative strategies to control mosquitoes are actually targeting only mosquito. So therefore, like the idea is to make mosquito healthy. So once it bites infected human and has parasite inside, so that parasite will kind of be blocked with those new control strategies. And therefore, another time when mosquito goes and bite another human, it will not transmit parasite.
Wow, that's really amazing. We have another question. Do you think that University in Banja Luka prepared you well for continuing your education? Also comment. Thank you for great webinar. You explained everything so well.
Thank you. I appreciate that. It really means a lot to me because I know that I was really trying to kind of like develop the slides that they will be understandable to everybody, because sometimes when I talk about it, I also don't understand. Thank you. I appreciate that.
Did University in Banja Luka prepare you well for the rest of your education, for a master's and your PhD?
Thank you, Jovana. Oh, I'm sorry. This is the question?
Yeah, that's the question.
Okay. Well, I mean, it's different. I cannot say that they prepared me or they didn't prepare me, because I was studying totally different things. Like I was mostly studying the main aspects of plant production in agriculture. So actually during all the course of my academic journey, I was kind of just switching from one topic to another. So I have to say that my switch from University of Banja Luka to master in Italy was actually pretty much challenging, because it was my first time actually to study in English. It was my first time to be away from home. Okay, beside that, but I mean, it was tough. It was tough, yeah. But anyway, I mean, those are just the small things that you easily overcome because you like what you do.
Okay, we have one more question. Does the presence of Wallachia, I hope I understand. Okay, Wallachia in mosquitoes cause no problems in the food chain?
No, definitely not, because Wolbachia is actually bacterium that is insect related, and actually it actually expresses its parasitism only toward mosquitoes and, for instance, like some nematodes that live. So they don't have any impact on the food chain. They're totally safe, because they are actually naturally occurring symbionts. It just happened that actually this particular mosquito doesn't have this bacteria, and this particular mosquito is one of the most, let's say, dangerous vectors of the viruses. And for that reason, scientists come up with the idea, okay, let's inject Wolbachia and it will show some phenotypes in this, and we can just like play around with that. So yeah, it's totally safe.
You get a comment. Thank you for your time and answers. If you have more questions, please ask. We are approaching to the end of the webinar, so interesting webinar, thank you. A comment from one of the...
Thank you, I appreciate it. I appreciate it.
I guess there are no more questions, but I will wait for 1 more minute.
Yes, of course.
Just not to miss any.
Well, mosquitoes are mostly boring, so I guess people just want to get rid of them, so.
Yeah, I think so too. They are annoying. And I'm glad that we don't have these type of mosquitoes here in Bosnia. We can handle those.
Yes, actually we don't have. I think we just have those, the regular domestic Culex mosquitoes, and I don't know, like what's
yeah,
there.
and we can't even handle those, so.
Yeah.
It's good that we don't have those dangerous ones. Okay, so no one is writing for now, and I think we should call it for tonight. Thank you so much, Jovana. It was an amazing webinar. I enjoyed it, and
Thank you, Ivana.
Thank you everyone for watching, and I hope to see you on other webinars too. This was really great, inspiring. The one thing that really left amazing impression on me is that you mentioned everyone that you think that
Thank you, Ivana.
Yeah, that's really great. And you have a comment from Tanis Tan. Great job, Jovana.
Yes, he's my friend from the lab. Thanks, Tanise.
Yeah, I'm so glad. One of the names you mentioned actually watched you too. I'm not sure which one now, but one of the names I connected, one of the names you mentioned watched it too. So that's really amazing. You have great friends and people who support you. And thank you so much for this.
Thanks, Ivana.
I hope to that our foundation will collaborate with you again. And thank you for your shared knowledge and for your time.
Thank you very much. Thanks to Bosnia and Herzegovina Future Foundations. Thanks again for allowing me to share my experience with mosquitoes with your members and with everybody who registered for this seminar. So thank you so much, and I hope we can collaborate again.
Yeah, it would be our honor. Thank you so much.
Thanks, Ivana.
Okay, good night,
I'll stop
everyone.
sharing.
Yeah, you can stop. Yeah.
Thank you. Thanks again.
I'm just removing the participants. Yeah, it was really great. When you started mentioning, at first I thought that you will mention just Biljana and that Italian woman.
Yes,
Zašto pričam na engleskom i dalje nije jasno.
Pa jeste Lalić, pa znaš kako, Biljana Lalić je stvarno, mislim, ona je baš, kako da ti kažem, ono, bila osoba koja je došla u pravo vrijeme i na pravo mjesto da me preporuči za taj master program, zato što je ona isto bila na tom programu i ne znam, ona kad je završila, prošlo je nekoliko godina, ja mislim 2-3 godine, i onda je pitala me Jovana da li ti želiš da ideš na master u Italiju, kao nisu imali nikoga iz Bosne već par godina posle mene,